Hi, my name is Robert Streiffer. I'm an associate professor of philosophy and bioethics at the University of Wisconsin-Madison, and I'm happy to join the Neuroethics Learning Collaborative today. I'm going to be talking about neural stem cells and neural chimeras and some of the science behind it, but mostly the ethics. Just to make sure we're on the same page about some of the terminology. So I'm going to be talking about the introduction of stem cells into non-human animals. Stem cells have two defining properties. One, they're self-renewing, so sometimes when they divide, they just replenish their own population. But stem cells are also capable of differentiating into several more specialized cell types. So they don't just reproduce themselves, they can also reproduce definitively specialized cell populations. Hematopoietic stem cells are stem cells that are found in bone marrow, and they replenish the blood supply and white blood cells, red blood cells, platelets. And so one of the stem cell treatments that's been around for some time now is the use of hematopoietic stem cells after ablation of somebody's bone marrow to replenish their blood supply in a way that would avoid problems with the previous bone marrow. So that's something that's been around for a while and doesn't raise a huge number of novel ethical issues. But in 1998, Jamie Thompson here at the University of Wisconsin was the first to successfully isolate human embryonic stem cells, and with these cell populations came the possibility not just of doing hematopoietic stem cell work, but pretty much any other kind of stem cell populations that are in the human body we can now produce or are working on learning how to produce with human embryonic stem cells, because of course these the special thing about human embryonic stem cells is that they are what's called pluripotent. They can produce any kind of tissue in the human body, whereas something like hematopoietic stem cells are restricted to a pretty narrow range. Obviously, more recently with work establishing induced pluripotent stem cells. Human embryonic stem cells are not the only cells that can do this now. Induced pluripotent stem cells can as well. And the issues that I'll be addressing with chimeras would come up equally with induced pluripotent stem cells, although historically speaking they were associated with human embryonic stem cells because at the time those were the only pluripotent stem cell populations available for humans. So with the development of human embryonic stem cell research, researchers became interested in exploring and using animals to explore the potential of these cells, and not just with hematopoietic stem cells, but with other cells as well. And many of these areas of research have a neural component to them. Or involve the central nervous system in some way. So a couple of quick examples that I'm familiar with. Su-Shun Zhang is a researcher here at the University of Wisconsin, and he figured out how to derive motor neurons from human embryonic stem cells. So he figured out what the relevant media conditions had to be and how to coax undifferentiated human embryonic stem cells along the pathway to becoming motor neurons. Along the way they became neural stem cells and then what are called progenitor cells, which are similar to stem cells in that they can produce many different kinds of specialized cells, but they don't reproduce their own populations like stem cells do, and then ultimately motor neurons. He then transplanted those into chicks, chick embryos, where their spinal cord was developing to see if the human embryonic stem cell derived motor neurons could in fact connect with the spinal cord in the appropriate way so that they could actually function. And this is important work for people who are interested in studying motor neuron degenerative conditions. So the most common one there, the most well-known one, is amyotrophic lateral sclerosis, or ALS, or Lou Gehrig's disease, where the motor neurons die and the result is essentially complete paralysis. Another example is work done by Fred Gage and his group at the Salk Institute for Biological Studies. He introduced undifferentiated human embryonic stem cells into mouse embryos, so mouse blastocysts, and then he tracked them, and he demonstrated that the undifferentiated human embryonic stem cells can in fact produce functional neurons inside living mice. And so he says that this kind of research permits, and I'm quoting here, the study of human neural development in a live environment, paving the way for the generation of new models of human neurodegenerative diseases and psychiatric diseases. So these are researchers who are interested in learning more about neurology, learning how to understand and treat neurodegenerative diseases, and they think that stem cells open up an important possibility here. As one of the main methods that they have for exploring this area is the creation of neural chimeras, where these are now animals that have some human cells introduced into them. And the idea of mixing human and animal cells raises several different kinds of ethical concerns. Some of them probably don't withstand much scrutiny. Others are more serious. It is interesting to note that there are several advisory bodies who have over the years issued reports on human embryonic stem cell research who seem perfectly fine, or at least, well, let's put it this way, they think that the objections that are out there to human embryonic stem cell research, the objections that focus primarily on the moral status of the embryo and the fact that you have to destroy an embryo in order to derive new cell lines, they think that those objections can be overcome. And these same groups often think that animal research, using animals as biomedical models, is also justifiable, and when done and according to existing regulations, is ethical. So you have these groups that agree that animal research can be done justifiably and human embryonic stem cell research can be done justifiably. But when you combine these two, by introducing or proposing to introduce human embryonic stem cells into an animal, they often call for some kind of special review. And this kind of research raises red flags, even for people who are okay with the underlying kinds of research, stem cell research and animal research. So it's interesting to note that even among sort of proponents of these areas of research, you still get some concern. A few of the most prominently discussed concerns here are the following. Some people object to this kind of research because they think that it's unnatural and results in an unnatural mixing of materials across species boundaries. This is the concern that is most widely discussed by bioethicists, but it's also usually the most quickly dismissed. I am, in fact, one of the few bioethicists, I think, who is actually in print defending this view. This was American Journal of Bioethics peer commentary in 2003 to a paper by Jason Robert and Francois Baylis, which was probably the first serious ethics paper on stem cell chimeras, and that was in AJOB in 2003. In the end, I don't think the argument works, but I think it's harder to say exactly where it goes wrong than many people think. And this debate about unnaturalness goes back, it has quite a history, it goes back at least to John Stuart Mill's essay called On Nature. And so there is this objection. Another objection, and this is the one that was not quite endorsed, but at least put forward for consideration by Robert and Baylis in the article I just mentioned, the AJOB article, and this is the idea that by blurring the species boundaries between animals and humans, the creation of chimeras could in fact cause people to rethink whether or not the boundary between humans and animals is really as sharp as is often supposed, and rethinking that may in fact not have good social consequences. There are institutions and regulations that sort of presuppose a pretty strict dividing line between animals and humans. So if you think about the regulations concerning what kinds of animals we can eat and how subjects have to be treated in research depending on whether or not they're human or animal, these rules are quite different. The human subjects protections are very different from the animal research regulations, and the rules seem to, the whole system, the whole infrastructure seems to presuppose that we can draw a sharp distinction here. And if we blur that, where we end up may not be as stable or as good a position as we're in now. So that's another objection that people have discussed. David DeGrazia, who has done a fair bit of work in animal ethics issues, has argued that because neural stem cell work in animals is not at a point where it could plausibly be claimed to be therapeutic for the animals in question, nor can it be claimed to be risk-free or even minimal risk. In fact, it's usually going to be quite harmful to the animals, that this kind of research is incompatible with the proper respect owed to great apes. He argues that great apes have, at least to a significant degree, many or most of the psychological capacities that go into personhood, and with those capacities comes a very high degree of moral status that would make these kinds of research impermissible. Depending on the view that one takes about traditional animal ethics issues and what it takes to have full moral status, some people might be inclined to extend his argument, not just from great apes, but also to monkeys, and perhaps, depending on the view, even mice. Obviously, this gets into large issues about the responsible and ethical use of animals in lots of other areas, because your view on this is going to have implications for whether or not other kinds of harmful, non-therapeutic research would be permissible with animals. Cynthia Cohen, Derek Vanderkooy, and colleagues have argued that chimeras, or at least a certain subset of neurochimeras, have the risk of violating human dignity. And this was an early article on chimeras in Nature Medicine, and then followed up with a longer article in the Kennedy Institute of Ethics Journal. And they argued that human dignity is grounded in certain kinds of psychological and emotive capacities, and that if the introduction of neural stem cells into a non-human animal were to confer those capacities without also at the same time providing those research subjects with the conditions that they need to actually exercise those capacities, that this would in fact be an affront to human dignity. The challenge for this kind of argument is to explain exactly what human dignity is and what kind of moral implications it has. It's a term that's tossed around somewhat cavalierly. It's used to explain what's wrong with everything from torture to not getting proper consent from human subjects. So there's a lot of work that needs to go into sort of conceptually clarifying what that concept is and what its moral implications are. And then finally, the final objection that I would mention is one that I've developed, and the idea here is that the regulations, as I mentioned, for proper treatment of research subjects depends to a very large extent on the species that the research subject belongs to. If you're a mouse, you get one set of protections. If you're monkeys, you get another. If you're a great ape, you get yet another. And if you're humans, you get yet another. And with humans, of course, we have a virtual prohibition on more than minimal risk, non-therapeutic research without consent, and that prohibition is absent from the animal research regulations. Whether or not you think that species itself is what matters here, or whether species is just a useful surrogate for other properties, the idea that you could have an individual that belongs to one species, say at the beginning of research, and then through the introduction of human stem cells, change that individual into a member of another species, or end up with an individual that has parts from different species, or end up with an individual that has an animal body but a brain that's composed largely of human material. On any of these views, there's at least the theoretical possibility that the creation of a chimera, if you introduced enough human material and if it had the right kinds of effects, could substantially increase the kinds of protections that were owed to that individual. And if the individual were then nonetheless treated in accordance with, say, the animal subjects regulations, then you would end up with something that was quite obviously unethical. You'd be treating an individual who has the same moral status as we do as if the animal regulations were sufficient to ensure ethical treatment of that individual, whereas of course the animal regs are woefully inadequate when it comes to the ethical treatment of human research subjects. So the idea here would be then that through the introduction of human stem cells, especially human neural stem cells, you could enhance the cognitive capacities of an animal so that it came to have the psychological properties that are sufficient for full moral status or personhood, and that would then render the subsequent research on that individual inappropriate. And the idea then would be that if researchers can't guarantee adequate treatment for the individual after it's been enhanced, then they actually shouldn't enhance it to begin with. So these are some of the objections that are out there. Different people obviously have taken different areas here under their wings and defended them differently. And there's a lot of disagreement right now about whether or not the kinds of outcomes that people are worrying about when they put forth these objections are realistic. There's a fair bit of data to show that introducing even a fair amount of human material into, say, a postnatal animal, especially an adult animal, would not actually change its behavior or psychological capacities very much. But there are nonetheless concerns that if you introduce enough of the cells, enough of the human cells, early enough in the animal's development, that they might in fact sort of dominate the developmental trajectory of the individual, and you could end up with an individual that was radically different from what it would have been had it just been brought to term normally. So there are also a variety of scientific questions then about the actual impact of the introduction of human materials into the animals, as well as, of course, ongoing disagreement about these ethical objections that I just mentioned. So I hope you find that helpful. If you're looking for more reading, there's a Stanford Encyclopedia of Philosophy article on chimeras that I authored that has a fairly comprehensive and up-to-date bibliography. So I'd point that to you for further materials. Thanks.